said hello, and the name of the talk is here again. This is the cupule Kannaskoppia, and I've been working on the Molteno Flora now for a long time, and my co-worker has been John Anderson, and he's actually joined us for this talk, but I see he has vanished. Anyway, John and I got married in 1968, and we did a lot of work on the Molteno. In 1988, we divorced, but we carried on working as research colleagues. And then in 2002, I married Keith Holmes and now live in Dorrigo, Australia. The Molteno Flora comes from this area of Southern Africa, which 230 million years ago was part of the enormous continent known as Gondwana. During that time, there are many other floras similar to the Molteno in South America, India, Antarctica, and Australia, New Zealand. These are all dominated by the Dicroidium leaf, which has a typical forked frond. Here is a geology map of Southern Africa, and you will see that most of the southern country is part of the Karoo Basin, which goes from Carboniferous to Jurassic. And this inner area is Triassic, and the Molteno is part of that. This map is based on Brian Turner's map. He did a regional study in the 1970s, and we were lucky to team up with him, and he helped point out some new localities for us. The first locality we ever worked on was Little Switzerland, and that is right up here in this top corner. The other geologist who did a lot of work on South African geology and on the Molteno in particular is Alex du Toit. He did so much research that he also collected plants from the Molteno and published a monograph nearly 100 years ago in 1927. His collection, he also illustrated himself with these beautiful drawings. My supervisor at university was Dr. Edna Plumstead, and she heard from another geologist about this locality, which was called Little Switzerland. So when I was a student, and John, she started me off on working on the well-preserved macroplants from this carbonaceous horizon, and John did a study of the spores and pollen from the same locality. After graduating, we both managed to get a job in Pretoria at the Botanical Research Institute, and we took our fossil collection from Johannesburg on loan. Over the years, we made many more collections, and in 2002, I retired after 24 years at that job, but I carried on working, and then John retired in 2008, and the collection was then relocated to Johannesburg, and the Pretoria collection was now taken on loan. There, the same cabinets were put into a compactor system. Another view of the lab, there's a compactor system in the back, and this shows the typical metal trays where our fossils are in. So it's very easy. You can take out a tray and you can immediately have a look at the fossils. Once John retired, all our other material went to his house in Pretoria. You can see the filing cabinets here in the back. And that's where we've now, in the last years, done most of the work. Another view of the same room. And here I have spent many hours. Every year I've been able to go back to South Africa to work on the fossils. A closer look at the Molteno outcrop. I'm going to show you this area in more detail, but we have 100 assemblages from all around, and we've numbered all these localities. And we've got, as you can see, many catalogued slabs from there. Little Switzerland is our first locality that we ever collected from. And another important one I'm going to mention is the Umkomaas locality, which is number 6. And you go all the way around the outcrop. And then this is our last one here. Okay, now you can read that for a moment. I'm going to have a little bit of water.
Okay.
Okay, I hope you've all read that.
Yeah.
All right, that was our first book. And then this is our second volume, which was on the other gymnosperm leaves. And we were supposed to then, in sequence, do the ferns. But because we found this special plant with attached leaves and fruit, we got sidetracked. And then we landed up doing the heyday of the gymnosperms, wherein we described all the fertile material from the Molteno. The unique discovery we called Kannaskoppia, and here you see the cupules attached and the leaves. Now, the word Kannaskoppia we found is a bit hard for English people because it's a local name. It was named after the locality called Kannaskop, and in Afrikaans, Kop is the name for a hill bearing canna plants. Now, these are not the monocots that are often grown in gardens. This is another plant called kanna, which the indigenous Khoisan people used to like to chew or smoke, and it now in the genus Sceletium. So Kannaskoppia was classified by us under the Ginkgo Opsida, which is an order promoted by Mayen in 1989. And a lot of other so-called seed ferns or plants with cupules are in this group. One of the most important ones for the Molteno is the genus Umkomaasia, created by Thomas in 1933. We now have found this from 75 assemblages. It consists of, we'll just show you that. So here's the write-up, and here is the original type specimen as Thomas described it. Here are some others that we have subsequently found. And this consists of a megasporophyll with paired cupules. And these paired cupules contain these beautiful seeds. This is from another locality where you can see the pedicel has swung down, but the cupule still got this recurved aspect. This is the type locality called, oops, I jumped. The type locality called Umkemas. And this is up in the Drakensberg Mountains. This is the fossil horizon here. And sometimes it was called the waterfall locality, because when there's a lot of rain, you get a waterfall here. Here I'm standing long ago on the oil shale, and this was described by the geologist I mentioned before, Jatoy. Here is John collecting at Umkemas, and we had to carry everything down the mountain. So these big blocks, we had to make some of them smaller. We now have 173 specimens of Umkemasia from this locality. Now, what happened is over the years, other people have called other cupules also Umkemasia. And recently we did a reassessment of this and reclassified some. I was busy reclassifying this one, which was called Umkemasia mongolica, when somebody else decided it belonged in the genus Doilia. Okay. Anyway, we finally got back to describing the ferns, which were published in 2008, and then the sphenophytes, which were e-published. So we don't have an actual book of those yet. But we were very happy last year. Whoa, jumping. We were very happy last year when on the 1st of December, our book about the Molteno Kannaskoppia was published. This is last year in July, when I helped John return all the Kannaskoppia fossils back to Johannesburg to put back in their place. Here you can see it's a large collection we've been studying. Here is the holotype of Kannaskoppia, and here is the photograph of the same and a close-up. And we want to thank Christian Autot for all the color photographs in the monograph. He has done a wonderful job with that. Here is the isotype, which happens to be on the same slab as the type. And it's an interesting specimen in that it shows how a lot of the preservation is in 3D. This little slab has been removed from this area. You see the two stars? So you can take this off, and underneath you can see more cupules and more leaves. On this side is a drawing with the flake removed and one with it on. Our reconstruction was based on 5 large attached specimens and about 50 megasporophylls. But strangely, we never found, except for the one time, we never found any more. And the female up to now is only known from that one assemblage. Here is the generic diagnosis. So it's a Ginkgo obstetrobulus with a forked axis bearing several megasporangia, which all consist of a single recurved cupule. And they're also all pedicellate. Now, about the same time or some years before, Taylor et al. In 1994 described the genus Petriella, and this was from a permineralized peat deposit in Antarctica. And this revealed amazing information. In the cupule, they had all the ovules. It's possible that Kannaskoppia and Petrielia are cogeneric, but what is known from one is not known from the other, so it's very difficult to decide. Here is a close-up of their Petrielia. Here's a cupule with ovules inside, another view of the cupule with 3 ovules, and another view. Now, it's quite interesting for us to try and surmise what lies in this cupule. We can imagine that maybe it's the same as what they have found in the Antarctic specimens. Now, not only do we have good examples of the female Kannaskoppia, we also have the male, which has been called Kannaskoppiensis. This is similar in that it is a forked structure, and it bears 2 rows of microsporophylls. These bear the microsporidia, and when they're young, well, immature rather, they are protected by a little operculum. And John is very good at dissecting and splitting open the rocks, and he's been able to chip away, and then this little operculum will suddenly jump off. So this is our reconstruction of it. And in the next one, I will show you an example. They're quite small. They're, I don't know what size it's on your screen, but this is only about a centimeter across. And some of these we were lucky enough to obtain a microsporangium still attached. So there's one still attached. This is the counterpart showing the microsporangium attached. We have made 8 species. I think I missed one. Yes, sorry, I missed that one. Here is another one. And in this case, they're all empty. There are no microsporangium left, but we have little scar marks. And we've interpreted these as being where the microsporangium were attached. We've made 8 species. That's the one I just showed you. Here is another. Here's another one attached. And here is a closer up showing it. There's a few of these strobili there, and they're attached to the stem. Another one here is attached to a stem with leaves. And these are also the same kind of leaves as you get with the female. So we are pretty sure that they belong. Here's a closer up with the leaves, the photographs. In this case, the male is not so clear. It's not so well preserved, but I'll show you the leaves. They're the same as we have in the female, and they show these typical anastomoses. So the leaves have been put in another genus. We initially called it Kannaskoppifolia, but more or less the same time, Herbst in South America was working on these leaves, and he made the new genus Rhacopterus. We thought we could keep this name for attached specimens, but Bonfleur decided otherwise. I'll just show you this again. So the veins divide, and then you get these anastomoses. They are irregular, and as you go up the leaf, the veins become finer. So these leaves have been known for a long time. They were previously described as chiropterous, and Retallick described them as Ginkgophytopsis. Also, other genera have been used. You'll have to read the book to find that. Okay, another picture. to show the irregular anastomoses and how the veins divide and eventually become quite fine near the tip. Now, we've described 12 species of the leaves. This is the one attached. This is another big one. This is the one I've just shown you. And some of them are very divided. This one, which is actually called Switzifolia, because it comes from the little Switzerland locality. I'll show it to you over here in more detail. And that's the Kannaskoppiensis that's associated. Now, we've described these leaf species according to what we regard as paleodemes. We first introduced this term in 1985, and I'll briefly go through it because it's quite important in our work. We regard a paleodeme as a fossil population, a collection of specimens, possibly representing a single breeding population, showing a unimodal distribution of variation, and, this is important, derived from a single fossil assemblage and geologically a discrete lithological unit. We use this paleodeme approach when we studied the Dicryoidium species. Here's an example of a slab from this locality. And here we have plotted all the different leaves that we found. You can see that some of them are very long, some of them are quite short, but most of them are in this area. And what's interesting, for every locality we have with Dicryoidium, you get a slightly different picture of the distribution. Now we get back to our Rhycipterous leaves. Here we have two assemblages, one from Umkamas and another one from Arsfoil. These are both placed in the same Rhycipterous species, but you will notice that the leaves, they look similar, but they vary, and they range from being quite small to some being quite big. And some here are also quite divided, but in general, they are not so very divided. Here, again, the same species, Rhycipterous rolleri, from another two assemblages. And it might be interesting for you that these were first described in South America as being Bayera leaves. They show these distinct anastomoses, and they are definitely not Bayera. Here is a paleodeme from the Umkamas locality. These were originally described by Du Toit in 1927, and I'll just read what he wrote. He said he had two leaves practically identical and set at an angle of 70 degrees, as in a whirl. And now we can agree with him and say they probably were. This is the largest leaf we have, quite large leaves. Here's a group of them. Here are some attached. And here is what it's groups of leaves. And we think these are all attached to the same stem. So they occur in separate little whorls along a stem. In the meantime, Bonfleur et al. In 2014 found Rhycipterous leaves attached in Antarctica. Here's a drawing of the same, and this shows the stem in more detail. Now, at present, the best record of leaves attached to stems comes from this Kannaskoppia recipterus plant. But there are also other good ones in Dicryidium, for instance. Here is one. And there is a photograph of the same specimen. Note the Dicryidium leaves here. We wrote a paper of that, published in 2008. And we added Lance Fitness, who was a collector. It was thanks to him that we found those. He showed me some, and then he showed me some of the big ones. Normally, he would not have collected this because he only collected pretty specimens. But I had told him that in science, it was important to also have all the other specimens that were not necessarily pretty, but were informative. I then sent him back to the locality, but sadly it was some weeks later and he couldn't find all the pieces. But he brought me all the bits that he could find, and eventually with the big jigsaw, we managed to put all these blocks together. And we found that there was a whole plant in this. It even has roots. And in the different layers, you get these whorls of Dicryidium leaves. Another one that we have attached is Phenobioera. This shows a stem with a branch and another branch. Now, we like to make whole plants, but except for when you get a whole plant preserved in something like volcanic ash, it's very rare in the fossil record. Most plants are dissociated, but we've been lucky with finding Kannaskoppia. But there are lots of questions. Is this a large part of a small plant or a portion of a large tree? What are the other clues? We've put together all the evidence we had from the Molteno and from Antarctica. We considered 6 points in detail. And you'll have to get the book and read about that there, because here we list it all. From Antarctica comes interesting information on the structure of the stem. And this supports the idea that these were pioneer plants with a slender woody stem to possibly about 1 meter in height. Here we've reconstructed them as pioneer plants growing along the edge here of the water. For all of these, we give a lot of extra information. For instance, here we have the recipterus, but we also add what other plants are available at that same assemblage. In this case, we have horsetails, 22%, and ferns. And these ferns are interesting because they are also whole plants. We've got the rhizomes and the whole leaf have been found. Further information we give is where the assemblage is at the locality. This particular one at Kannaskoppia is actually a road cutting. We did try and go back and get more, but although we got a big excavator, we never found any more there. We also add what insects are around. At this locality, we have found no insects, but we have evidence of insects being around by insect damage of the recipterus leaves. In the Molteno, we have recognized 7 habitats. One of the important ones is the riverine habitat growing along the river. And I'll show you the last one, which is our fern Kannaskoppia meadow. And I'll show that in more detail. Here we have the fern Kannaskoppia meadow, which we think was possibly part of this braided river system. In the more mature area, you get the river-round forests, which is the Little Switzerland and Umkamasia site belong to that. Another one I can point out is the lake deposits around some of the lakes. So for each of the 12 whole plants we have, a habitat page is provided. We have the reconstruction and all the other extra details and show where the specimens were collected from. Show that in more detail. Here's a big hole we made at this particular locality. And here's the detail of the habitat reconstruction. Here are the leaves and the male. And in this particular case, because it's also the river run, we have a big dicroidium tree. We have we still recognize these as being pioneer plants near water. And here we've got some growing a little bit further away. This is another one in the Sphenobiera woodland, habitat 4. Here is our reconstruction with the male. And here are the Sphenobiera leaves. And these more autumnal leaf trees are the Sphenobiera trees. Another locality which fits in the Sphenobiera woodland is Bird's River. But interestingly enough, we have not found any Kannaskoppia or Rhacopterus leaves at this locality, even though we have a large collection, over 2,000 slabs. But there are a lot of good insects from them. Another habitat is the Hardiphyllum thicket. Here are the Hardiphyllum trees. And in this case, we do not have a male. So it's just the Rhacopterus plant that we have reconstructed here. So that's growing possibly in sort of swampy ephemeral places. Oops, I went too fast. And there we think that this deposit was near a little lake like that. This is from Antarctica, where Bonfleur et al. Have reconstructed their plant. It looks very big here, but it's actually quite small because the artist has given you the view as if you're a little insect sitting here and you're looking out, up into the sky, into the night sky, and you can even see the southern lights. So these are his reconstruction of these plants. Just to remind you again of Petrielia, which has got the exquisite preservation. And it was suggested that maybe some of these early cupules may be sister groups to the angiosperms. So here in our phylogenetic chart, we have the Molteno here at 230 million years approximately. And we've placed the Petrielales as possibly some sort of sister group to early angiosperms. It's interesting that now, more recently, there's a lot more interest in these Mesozoic cupules, as demonstrated by this paper in Nature, May 2021. And two of the authors, Fabio Herrera and Peter Crane, came to visit us in Johannesburg. There's Peter Crane and there's Fabio. And they are now studying another cupule from the Molteno. And we hope to have that published in about a year or so. While they were visiting the ESI, the director, Marion Bamford, showed them the other things at ESI, the famous fossil man bones. And here we are also, there's John again, and here we are studying some of the new cupules that we will describe. So, come to concluding words. Okay, I've discussed something of what we've covered in the Kannaskoppia book about the female cupule and the male microsporangium and the leaf species, and given you some idea of our approach to the reconstruction of whole plants in the Molteno habitats. The cupules, Kannaskoppia and Petrielia, trigger further questions as to an angiosperm origin among the seed plants of the Triassic. Furthermore, this comprehensive study is a model of what can be done for some other fossil plants. So, thank you.
Thank you, Heidi, for your brilliant talk. So informative and so perfectly illustrated. Thank you. And now we can pose questions. If anybody wants to pose it in your own voice, switch on your phone, raise your hand and ask. Bruce, I see your hand rising. Could you switch on your microphone?
Switched on.
Okay.
Now
Yeah.
you can hear me. I'm curious as to where or what would be a convenient source for all of those books that were shown. Is there any outlet?
Well, yes, I have to confess that it is a problem, and we are a bit guilty about that, because the original two books were published by Balkamer, and sadly, he just wanted to make money out of them, and he never sold very many because they were expensive. And because they were expensive, we never thought of buying them back. So they are now out of print, and Balkamer eventually closed down the business. So except via Amazon or somewhere, they are not available. But they have now been, some of them have been put on, or e-published, or are available. And I can, if you give me your email and that, I can send you a link to where you can get some. So some are available digitally now. And we are thinking that maybe we must just, the ones that are not available, maybe we must just make them available digitally now, because I don't know, it's a bit of a problem when you have something officially published through a publisher. But the more recent books, the ones we did in Pretoria, some of those you can still buy from the bookshop at the Pretoria Botanical Institute. It's now got a different name, but I can send you the link for that. And as I say, the last two books are both e-published and freely available.
Thank you.
Thank you. And I see email from Bruce to Heidi in the chat. So Heidi, you may copy Bruce emails from the chat. And
Okay.
I see.
Will I be able to see that afterwards? Yes.
Yeah.
I
And
see it now.
There are several questions and one comment in the chat. Svetlana Polevova found, I suppose, a type mistake in your conclusion. Male megasporangium. Yeah, there's something.
Megasporangium. Oh, yes, it should be macro. Oh, la la. Well spotted. Sorry. I didn't have anybody to proofread. It's well spotted. It must be macro. Yes. Thank you.
Macro.
Micro.
Micro. Okay. And the question from Evgeny Karasov, also in the chat. You may read it and I read it for everybody. Heidi, what are the main differences between the female organs oncomeseles, petrileles, oh, it's so difficult to read Latin, and Kannaskoppeles? What are the main differences between them?
Okay, so the main differences between the Kannaskoppiales and the Umcamassiales. And what was the third one? I missed that.
Petrileles.
Petriales. Oh, all right. Well, the Petriales are only preserved, you know, they are only preserved in peat. So they don't really have a good idea of the 3D structure on a macro scale. On a micro scale, they know all the details, but exactly how these cupules fit together, they are not sure, but they have made some suggestions. But of course, on a micro scale, they have all the details. And the problem is, for us in the Molteno, we have the cupules, but we don't have any structure preserved. We don't have cuticle from that locality, and we don't have any way of finding out anything more. What I showed in the photographs, that's all that you can see. And now he wanted to know what the differences were with Umcamassia as well. Is that right?
Yeah.
Okay. Well, the basic cupule is somewhat similar, but in Umcamassia, it seems there's only one ovule or one seed. If there's more, then possibly they don't reach maturity. But so far, we've only ever found one seed per cupule, while if in Petriella, they seem to have quite a lot. They've got, 6 or 7 or maybe more sometimes. So that's one difference, that they've got a lot more ovules. Otherwise, it's possible that the basic cupule might be similar. And I think that's true of other plants around, even like Doilia and that the sort of basic cupule structure is superficially similar in a lot of these. Was this another part to the question I've missed?
No.
Well,
Thank you. Thank you.
Is that good?
we have more questions also in the chat. The next one from Natalia Nosova. She is writing, thank you very much for this very interesting report and for the link to the PDF of your last book. Did you compare Kannaskoppia with Carcania and Yarudia, she and the authors? What are the main differences between them? So comparison with Carcania and Yarudia.
Okay, no Carcania. I'm not sure what your Rudia is. I forget that one. Okay, in details, we've made no big comparisons. I know that we did do some comparison. I can just quickly check what we wrote before. 1 minute.
And meantime, I'd like to remind that you may pose your question in your own voice, if you prefer.
No. We did not compare it closely to Carcania. We did compare it to Catonia, which is the cupulate structure from the Jurassic of England and other places. And then there was also some similarity with what's called polyspermophyllum from the Permian of Argentina. There's also a cupulate structure there that has some similarity. So I think that partly answers her question. So maybe somebody should do a better comparison with Coccinia.
Natalia Nosova, do you have any comments on this? Or just thanks? Well, thank you, Heidi. The next question from Evgeny or Tatiana?
Tatiana.
Yeah, it's me.
Tatiana.
Yeah, thank you very much for your talk. Could you please show a slide about paleodemes, about fossil populations?
You'd like to see that one again? Yes,
Yeah.
sure. Okay, it's quite far up. There, that one, yes.
Oh,
Yes. I wanted to ask, how can we understand how much time took it to form a lithological unit from which we take a paleodeme?
Yes, that we admit that that is difficult. We have some of our lake deposits we think are quite long because you can almost see almost like in glacial times, you can see the lines. So each time there was a big flood, each time there was a flood, there's like another layer. And it's very hard to know, was that a flood once a year? Or was that a flood once 100 years? Or was it a flood once a month? So some things are quite difficult to decide on. And also sometimes we'll get an assemblage and it could be going for a long distance. So what we do is we say that we try and restrict an assemblage to within 100-meter area. Quite often our localities, we're not able to get much more than that anyway. But it is a problem. And then so unless you can get absolute dating, which they're getting more clever at doing that, we will never really know. And some of our localities seem to be like an oxbow little lake or channel that's filled in. And again, by comparison with modern times, you can say it takes maybe 100 or 1,000 years. But again, we don't really know. So there's always a lot of guesswork. But at least we are trying to get more a fossil population. Most people, or they used to, get leaves from different places and lots of different times. And then they call them all one species. And we're just trying to zero in and get something that's more likely has come from one area and one time. But of course, it's not perfect. Does that partly answer your question?
Tatiana?
Yes, thank you.
Thank you. We see a comment from John Marshall in the chat. Many thanks. Excellent to see how all this has developed across the decades. Got to go to another meeting. So,
Okay.
and thanks from Natalia Nosova for your answer. And I don't see if we have more questions. Well, if not, I'd like to pose my own. How about pollen in the pollen organs?
Oh, well, again, in the localities that we have the Kannaskoppiensis, the male, most of our Molteno localities do not have cuticle or pollen or spores, sadly. The one that has about the best is our first locality, Little Switzerland. And John has done a big study on that, but many, many years ago, and he never finished it. He has got all the photographs, but he never wrote them up. It became very complicated to get all the names for the spores and pollens. And from Little Switzerland, we do have some preserved Kannaskoppiensis, and maybe it's possible to get pollen out of that. But we do not have many specimens, and we have not tried that. That again is a bit like a specialized study to do that. So in the Molteno, I must tell everybody, there is still a lot of potential for a lot more work. And we've got other pollen organs where also it would be good to investigate whether one could get pollen from them and then link it to, yes, the pollen and spores that are known.
Yeah, it seems that they're a good addition to your brilliant paper, to your study. And perhaps John would like to add anything about unfinished and not published work? No?
Are you there, John? I don't think he can hear you. Can you hear John?
Perhaps not. Okay.
He doesn't hear. Yes. But I think sadly now he will never still do that. But if there is some student somewhere, maybe they will. Oh, here's John. John, can you hear what we're talking about?
I can hear you, Heidi. Yes. Can you hear me?
The question? Did you hear the question?
It was just for me to comment further, I think. But can anybody hear me now?
Yeah, we hear you.
We can hear you.
And we are curious to read the paper Heidi mentioned as as unfinished paper.
Yes, way back in the 1960s, I worked on the palynology of one or two localities, Little Switzerland, for instance. But never since. I've been working on the macro paleontology since then as with Heidi. So we would greatly welcome a couple of students to get involved in the palynology. That is certainly something that needs to be done, and we would very much welcome somebody. And we still have about 10 years or so of work on the Molteno to do to finish various other volumes. We've got 4 more volumes to finish. The final one will be a nice synthesis of the entire flora. We've got to stay fit and healthy for the next 10 years to do all that. But we're very, very eagerly welcoming anybody to get involved with us.
Thank you. Well, I see several more comments in the chat from Bruce Cornett. Thank you, Heidi, for a most comprehensive presentation. Many thanks from Ivan Savinov, and also thanks from Gwinar, from Gitan Gwinar. And well, no questions, just thanks. Well, I'd like to pose one more question. I saw in one of your slides that you first examined the uppermost level of the rock sample, take off this level and examine something underneath. So what do you think, will it work if you study your material with tomography?
Oh, well, that's quite yes. You're referring to this slab where we had 2 different layers.
Yeah, this one.
Yes, well, actually, when Peter
right.
Crane came in 2022, he actually put some specimens aside which we hope to study via tomography. But I think we're still in the queue for that work to be done. And of course, most of it you can only do with quite small samples, but we're hoping to get something like that done. But I have to just add again that this locality, this Kannaskoppia locality with these Kannaskoppia plants is a bit unusual in that in most of our localities, the rock is very nicely layered and it's easy to split. But in this one, it's not so easy to split. But when you bang the rock, it usually will split where there is something. And we think that this was laid down very quickly. There was some sort of a flash flood or an overbank spill that enclosed these plants very quickly.
Thank you. Well, I do not see more questions either in the chat or with hands. So have we finished with the discussion? It seems so. Thank you, Heidi, for your brilliant talk. Thank you, our listeners, for attending our meeting, and I hope to see you next time.
Good.
Thank you very much.
And thank you, Natalia, for chairing this. Very nicely chaired. Thank you.
Thank you for arranging this.
Heidi.
It was lovely to see everybody.
Yes, no, thank you for coming, and it was a pleasure to do this.
Well
It's
done,
very
Heidi. Well
comfortable
done.
way, a very comfortable way of giving a lecture. I don't have to buy an expensive plane ticket and I can sit at home and do it. Very nice.
Yeah, thanks again, Heidi. Thanks, Natalia. Thanks, everyone.
Yes, thank you, Natalia.
Thank you.
Okay.